Like to welcome everybody to the UK Beef webinar. It is our distinct pleasure to have Dr. George Perry with us tonight. Dr. Perry, Jeff, and Darren and I have known each other for literally decades now. And some of us have ties to the great state of Missouri. We'll leave Bullock out on that one. But anyhow, Dr. Perry was raised in South Central Texas on a small beef cattle operation. He received his BS in animal science from Texas A&M. Then he obtained his master's and PhD in reproductive physiology at the University of Missouri, where he spent a large portion of his time up at the USDA Research Center in Miles City, Montana. Dr. Perry was a faculty member at South Dakota State in the animal sciences department for almost 20 years. And I was commenting with Dr. Perry earlier that it just time flies. I didn't realize he was there that long. A few years ago, he got a new job back home and is serving as a professor at Texas A&M AgriLife at the Overton Research Center. His research areas are vast and include estrus synchronization protocols. He's developed several on his own: follicle growth and regulation, pregnancy success, embryo mortality, vaccinations and the impact of the immune system on the reproductive system. And for, I guess, George, you didn't want to, you wanted more things to do. You wanted to look at sperm longevity and fertility. So he does both male and female reproduction. I'm not exaggerating at all, and that Dr. Perry is among the top 2 or 3 beef reproductive physiologists in the world, and we are extremely happy to have him here. And with that, George, take it away.
Thank you very much. It's an honor to get to talk to you all this evening, and to kind of culminate all of that, our research really focuses on the beef herd management and how it affects reproductive efficiency. And so, yeah, we've dove into all different areas. And what I want to do tonight is kind of talk about those different management practices and how they can impact reproductive efficiency. And when we look at things, I like starting off with this slide, and Dr. Cushman at the USDA Meat Animal Research Center put this together a few years ago. And to me, it really drives home what management does for reproduction. If we look at this, these are female reproductive traits when they're expressed early in life to later in life. So we go everything from things you can count soon after birth on antral follicle count, age at puberty, tract scoring, uterine horn diameter. Here's heifer pregnancy rate. We can see all the way out to mature cow pregnancy rate. And what we see is the heritability of all of those decreases. And you may ask, well, why is it decreasing? Because what's really the difference between heifer pregnancy rate and pregnancy rate in your mature cows? Well, if we think about all of these things as we get later on in life, management has a chance to mess them up. And so our management can actually trump the genetics that are there, and we can see its overriding power in things as we get later on in life. And so what I want to do tonight is actually go through, and I've set this up through the reproductive cycle. You know, what are some things that occur before the start of the breeding season, during the breeding season, and then briefly talk about the male at the end. And so one of the things Les mentioned that we've worked on for several years now is actually vaccines. And as he mentioned, I'm a reproductive physiologist by training, and started doing a lot of collaborative work on vaccines and how they could impact reproductive performance. And it has been a true learning experience for me to go through this and to learn what we know about vaccines. Because when when a company goes to license a new vaccine, they have to show efficacy of that vaccine, which means it's going to protect that animal or shows a antibody response when that animal is exposed to the virus or pathogen they're being vaccinated against. But a criteria that's not in there is some of the side effects that happen in the body. And so when we think about it, we can go back many years. Here we're seeing in 1986, when animals were experimentally given IBR, we can see a normal corpus luteum up here. And I've got another picture a little later that'll show things a little bit more in detail. But we can see nice large luteal cells producing progesterone. When we get down to these bottom two slides, we can see mononuclear cell invasions, that's the dark stained cells. We can see necrotic tissues, necrotic lesions in the corpus luteum. And so we can see that when an animal gets infected with IBR, these negative consequences that occur. Well, what's interesting is starting in about the 90s, people noticed that when using commercial vaccines, they were seeing some of the same effects. So these ovaries were collected 9 days after vaccination. And if we look, we can see lymphocyte invasion, so the immune system actually invading into the CL. We can see large areas of necrosis. And so this was occurring both with vaccines. And so where I really started getting into it was we started getting asked, what impact does vaccination have on my replacement heifer program? And when I was in South Dakota, there's tremendous variation in where people's replacement heifers were coming from. Everywhere from people, purebred operations raising their own, to commercial producers going to the sale barn, and what was going through the sale that day was their replacement heifer selection. And so we started asking, what does vaccinating do to reproductive efficiency? And really, if we vaccinate at the start of a synchronization protocol, do we have negative consequences? And so if we look here, abnormal cycles are what happens when a corpus luteum regresses early. So that animal can ovulate, she can get bred by AI or by natural service, but if that CL regresses early, there's no way for her to become pregnant. And if we look, our control animals, nobody had an abnormal cycle. Modified live group, about 40% had an abnormal cycle. Our killed vaccines, either one dose at the start of the synchronization, or a dose at the start of synchronization and a dose 28 days earlier, we can see about 10 to 15% abnormal cycles. So looking at what this is, this green line is progesterone profiles that we would see in our control group. If we look down here, all of these red dots were the modified live vaccine, and we can see they're actually regressing their CLs early. And so, as I said, there's no ways for those animals to truly become pregnant. And that's exactly what we saw. When we vaccinated animals, and these were naive animals in this study, at the start of a synchronization protocol with a modified live vaccine, our conception rates ran just over 30%. Compare that to our control group, where we're up around 90%. Our killed vaccines sitting 70 to 80%. Now, like I said, we had known some of this was going on from years before when animals were infected. Now, what we did different in this study was we actually tracked these animals to see, okay, if they have an abnormal cycle, when do they breed back? And so these animals had every opportunity possible to get bred. They were synchronized. We actually double AI'd them. We turned out bulls with them. So if they were in heat, they should have been able to get bred. If we look, that second service, our killed vaccines were back up to good fertility. Our modified live, we're still sitting at 30 to 40%. And so what that really did was impact the dogma that was out there on how vaccines can truly impact reproductive efficiency. Because it had been known, as I pointed out, we give a viral vaccine, a modified live vaccine, it can impact this dominant follicle and the corpus luteum that forms from it. But when we think about the synchronization protocols that we're using today, what do we do at the start of all of these protocols? We're giving an injection of GnRH. cause this follicle to ovulate. But most of the time, we don't care about the CL that forms because we're putting in a cedar or a progestin to keep those animals out of estrus. So what we're interested in is this ovulation here. But what we were able to see is when we vaccinated these animals at time of this GnRH injection with a modified live vaccine, we ran about 30% conception rates here and just over 30% out here. And so not only are we affecting this dominant follicle, but we're affecting the smaller growing follicles all the way out for up to about a month after the breeding season or after the vaccination during the breeding season. And so when we start thinking about these things, we know follicular growth actually occurs in wave-like patterns. And we know this egg that we're trying to get to ovulate at the start of the breeding season or during the breeding season has actually started to grow somewhere between 42 and 90 days earlier. And so when we're talking about naive animals that have never been vaccinated before, we know that modified live vaccines can have a detrimental effect. And so this was really the first study we dove into because we were thinking that the synchronization protocol would overcome the impacts on that dominant follicle. So after this study, we started getting lots of questions on, well, what about previously vaccinated animals? You know, surely if they've been vaccinated before, we don't see the same consequences. And when I was a graduate student, there were two papers. This one was after I was a graduate student, but for years, we always referenced these two papers on using modified lives in previously vaccinated animals. So here they had almost 800 animals. They had 3 doses of a modified live, with the last one being either 40 or 3 days before the peak of the breeding. And if we look, no difference in conception rates, no difference in days to conception. Earlier, Dr. Stormshack had actually gone through, this was a herd in Kansas, just shy of 300 heifers. They were vaccinated either 30 or 9 days pre-breeding. No difference in estrus expression, no difference in AI pregnancy rate, no difference in breeding season pregnancy rate. And so we always talked about these two papers to producers and saying, oh, everything is fine. Well, after that first study, we started digging through the literature and looking at what had been published. Because what stands out in these two studies is they're comparing intervals, 30 versus 9 days, 40 versus 3 days. They're not comparing it to a control group that never saw the vaccine. And so as we started digging through the literature, we found a couple of papers. Here's 40 and 10 days pre-breeding, 61 and 31, but they used a modified live or a killed vaccine. And if we look, 20% difference in conception rates, 15% difference in conception rates. Statistically, in the manuscript, they say they're not different because there was only 10 to 20 head per treatment group. We follow that up with another study that came out later. There's 60 head in the vaccinated group, 20% difference, 20% lower breeding season pregnancy rates compared to saline. And so Russ Daly, who was our extension veterinarian there at South Dakota, and I were there, okay, if we're going to address this question, what do we need to look at to truly get an answer? And so the first study we put together, we wanted to look at a modified live, control, and saline in enough animals. And so there are about 1,500 head in this study. There's about 500 head per treatment. We did this in herds in North and South Dakota. All of them had been previously vaccinated. A lot of the herds were herds we had worked with for years, and so we knew their vaccination history. They got Bovishield, either saline or virus shield, exactly 30 days before the start of the breeding season. So right by label. The virus shield group actually got 2 doses because they had never gotten virus shield before. And so to follow label, they got a dose 30 days before the breeding season and another dose 30 days earlier. The saline group got saline, both injections. The Bovishield group, because most of these herds or all of these herds had been using Bovishield, got saline the first one and then Bovishield 30 days before the start of the breeding season. So everybody was in the same pastures within a farm and was managed the same way. So what did we see? We can see about a 5 to 8% decrease in AI conception rates in the modified live group compared to the killed vaccine group. Our control animals are intermediate. We came back and looked at a preg check 56 days later after AI. So why is 56 days important? Because at 56 days, can we not only look at the AI conception rate, but we look at that first return to estrus and how well was the bull able to pick up the animals that cycled back. And we can see here actually a significant decrease in the modified live group that carried all the way through the breeding season. And so when we're looking at that, we actually saw this decrease at about 5 to 8% across the herds. And this had everything from replacement heifers to cows that were about 13 years old. All of them had had multiple doses of modified live. This study actually created a lot of controversy. People were wondering, okay, what's going on here? We followed it up with a second study looking at, again, modified live versus cattle master, which is a chemically inactivated vaccine. It's kind of a unique vaccine, so it's classified as a killed vaccine because the IBR will not replicate at body temperature. Again, about 1,500 head, and we can see a significant difference. Again, it's about that 5 to 8% difference. At the end of the breeding season, there was not a difference. Now, what was different in this one is we worked with cooperator herds. We worked with cooperator herds, and we came in with 1 dose of either one of the vaccines when they normally vaccinated. And so these cooperators, we have cattle that were vaccinated somewhere between 28 days and 90 days pre-breeding. And so there was quite a range in there, but we see the same thing. So when we finished these 2 studies, I used to tell people, okay, I'm confident something is going on. We've got 18 different herds. We've got 3,000 head, and we're seeing this difference of 5 to 8% consistently between modified live and killed vaccines. So what we've really done since then is try to follow up on what is going on. Because in these cattle that have been previously vaccinated, there is a paper out there that showed there was no necrotic tissue in the ovary. They gave 2 doses of a killed vaccine. They came back, they vaccinated them with a modified live. They overectomized them, and they saw no necrotic tissue. And so it was kind of, okay, the necrosis isn't going on. What can be occurring? So I had a student just finish up this past spring, Caitlin Epperson, and so both her master's and PhD focused a lot on the impact of vaccine. And the first study of her PhD was really looking at what could be impacted. And so we took a group of cows that had been previously vaccinated with modified lives. We set them up, we synchronized them so we knew the stage of the cycle. We vaccinated them. And in this group of animal, 24% of the group vaccinated with a modified live had an abnormal estrus cycle. None of them vaccinated with a killed vaccine had an abnormal cycle. And these were all mature postpartum cows having normal cycles before the study ever started. 21% of those actually regressed their CL early. 3% had a delayed rise in progesterone after ovulation. What really stood out is we were taking blood samples every other day. And if we look at all animals, as you would expect, progesterone was lower in the modified live group. But what's interesting is if we pulled out those animals, this 24%, there is still a big difference in progesterone concentrations between those that got a modified live or a killed vaccine. And if we graph it out over time, we can see this separation in the progesterone profiles. And so what does this decrease in progesterone, what's actually causing this? And so we did a study where we actually came in and we overectomized animals. And just like what's been reported before, we never saw any necrotic tissue. But if you look at these 4 pictures, there's some very obvious differences. If we look at this control group, we see some nice, healthy, large luteal cells here, which is what's producing the progesterone from that corpus luteum. If we look at our killed vaccine, we can still see all of these extra cells that migrate in, but we still see our healthy large luteal cells. When we come down to these 2 here that are from modified live animals, their CLs look totally different. We can see this white frothy area. We really thought these cells were dying. We've actually looked at it and they're not actually dying, but they're not being very productive. And so when we actually looked at percentages, when we looked at large luteal cell numbers, we can see both the control and killed animals had similar numbers of large luteal cells per section of the CL, which was more than what the modified live animals had. We can actually see the killed had more total cells per section. So when we look at it, the controls actually had the largest percentage of large luteal cells. This is where we're getting our progesterone from. That progesterone is needed for preparing the uterus for that embryo. So we need that progesterone production. And we can see here when we vaccinate at the start of a synchronization protocol, how we can negatively have impacts on that progesterone production. But it really still didn't get down to why. And so what we've been doing more lately is digging into some of the mechanisms of what can actually be going on. And so lately, we went back, took some of the blood samples, and looked at cytokines. And so the cytokines really signal inflammation throughout the body. And what's interesting is we know some of these, interferon gamma specifically, can have direct impacts on luteal function. And so when we think about that, we can see there's big differences here between our killed and our modified live animals in the amount of interferon gamma present. We can see some others here between our abnormal and normal cycling animals that we actually think this negative effect is through a whole upregulation of the immune system. And the fact when we get to a modified live and you put that virus into the animal and it starts replicating, you upregulate the immune system so much that we're having negative consequences on reproductive function. And so it's not a permanent effect. Depending on when an animal is vaccinated, they do recover from it. And so, as I mentioned in that one study where we worked with producers, we ranged from 27 to 90 days pre-breeding. And what's interesting is the killed vaccine was always higher than the modified live, but even in the killed vaccine, we saw an increase in pregnancy success when we got out past 45 days. And so it didn't matter which vaccine we used, we saw those benefits as we got out further. Now, as a reproductive physiologist, that makes sense to me. We can go back to the 1980s and we know the virus can impact this large dominant follicle. We know it can get into this follicle, it can affect the cells in it. And so if we think back, this antrum starts forming about 42 days before an animal ovulates that follicle. And so if the virus can get into this large antrum, why wouldn't it be able to get into these smaller ones? And so we see this process, and when we get out past 45 days, all we have are these small follicles that don't seem to be impacted by vaccine as much. And so that really gives us a time course that we need to get vaccines into animals early. Now, one of the things that makes that complicated is when should I give them? And I always put it as pre-breeding vaccines because we think about pre-breeding vaccines to keep the animal's immunity the highest during the breeding season. And when we think about the cow herd, on why we vaccinate the cow herd, it's for reproductive performance. We're not as worried about growth. We're not worried about as finishing. We want to keep the cow herd healthy for reproduction. And so the first thing is replacement heifers, we need to start building their immunity. We really need to get them vaccinated before or at weaning and start building that immune system into them. Both cows and heifers, from the data we've shown, we need to be at least 45 days pre-breeding to give them that chance to get over that upregulation of the immune system. And if they're naive animals, we need to be really cautious and not even use modified lives near the start of the breeding season. Now, if we're using killed vaccines, we have a lot more flexibility because we know killed vaccines are also safe in pregnant animals. And so it gives us that flexibility, but we get modified lives into them at weaning to start priming one side of the immune system. And then we can get killed vaccines into them as they enter the cow herd to prime the other side of the immune system. Now, when we start talking at least 45 days pre-breeding, now we get into seasonal effects. And so as I moved here to Texas, that was one of the questions that started coming up is, well, is there a difference if I vaccinate in the summer or the spring or the fall? Does it matter? And so Sarah Blasky is a new master student who just started this past January with me. Her first project was to actually look at that. And so we came in, we vaccinated animals either in July or in November. And so apologize, these are still in Celsius, but we're talking 100-degree weather for July temperatures. We're talking 70-degree weather for November temperatures. We collected a lot of blood samples to then see, was there a difference in how these animals responded depending upon the season of the year? Just looking at the viral side, so BVD and IBR. If we look at antibody production, there's no difference. Here's pre-vaccination, post-vaccination, pre-vaccination, post-vaccination, no differences in the antibody responses. And so when I talk about the killed and the modified live vaccine and why we need to get one in early in life and the other one later in life, is we actually have two parts of the immune system. We have antibody production, and then we also have all of our different immune cells. And I'm not going to go through all the different immune cells. We actually have a flow assay set up that we can measure several different cells, our natural killer cells, our activated T cells, antigen presenting cells. And so what I want to show you here is two of those. These are antigen presenting cells. When we vaccinated in the fall versus the summer, we see one effect on one cell type. We see the opposite effect on a different cell type. And so one of the directions we want to go with this is depending on where we're getting into, what cell types are more important. And so we need to make sure that we're addressing the immune system if we're relying on it to have immunity for an animal that's exposed, that we're having the best immunity we can. And it's not always just measured by antibody production, because antibodies are there, they do protect, but that's only one part of the immune system. And so when we're thinking about that, we want to protect animals. And are vaccinated animals totally protected? And this was actually where Caitlin, during her master's, really got started on the immune side, was a study we were doing on synchronization. And we had some herds that just totally crashed. And we were trying to figure out what the problem was. And so we started looking into the immune response. And so we see here, if the neighbor's animals are actually infected, this is showing BVD, we can have a cow here that has a good immune system, but depending on when the exposure occurs, we can have a calf born that is a PI calf, and then can actually have consequences on the rest of the herd. And an example of that was a producer here in Texas sent me this picture this summer. Now, I don't know how well y'all can see some of this standing out, but this is his fence line. This is one of the neighbor's cows. Now, what stands out in the picture to me, knowing the situation he was in, was actually, here's 4 different carcasses laying in the neighbor's pasture. This is a totally different risk management situation than if you're in a herd with different neighbors. And so he knows he's getting exposed to something from his neighbors. And that's exactly what Caitlin saw in her master's project. These were the herds that we used. We had 9 herds. We can see every one of them was vaccinated. There's both modified live and killed vaccines. All of them were 30 to 45 days pre-breeding. And we went through, and when we preg checked animals, we had herds that had horrible conception rates. And so fortunately, we had the blood samples. We went back and tested all of them for the antigen capture assay for BVD. And so what does that tell us? It tells us that the animal had an active infection of BVD going on. And so the herds, not individual animals, but the herds that had one positive animal or more, which there was 4 of the herds that had at least one positive animal on day 21 of the breeding season that were positive for BVD. When we looked at those animals, we're running about a 20% decrease in conception rates in those herds compared to the herds that were not exposed. What's interesting, second service, because this was a resynchronization study, we're at 27 or 17% difference between the exposed herds and the non-exposed herds, 2 rounds of AI, and a 20% difference for the entire breeding season. So what was interesting about this is we had a single blood sample on day 21 of the breeding season. We know these herds had an active infection. When we came back after the breeding season, there was not a single animal that was a PI. So what does that mean? That means these animals had exposure, and we know where the exposure came from. There were some fence line contact with cows that came from a sale barn. These animals themselves weren't persistently infected. They were vaccinated. The vaccines protected them from getting very sick, but we still see that transient infection going on. And so when we saw that transient infection going on, we see that decrease in conception rates. 2 of the 4 herds we were able to track the calves out of. There was not a single calf born that was a PI. And so the vaccines did their job, but we see this drop in conception rate. And actually, we're just working with a herd here in Texas that had a similar situation occur, but they purchased animals from a registered sale. The cows they purchased were negative, but the calves that were born, 2 of the 6 calves from the cows they purchased were born as PI calves. And unfortunately, we don't have a test for testing that calf that's in utero. And so we really need to think about the exposure level of our herds and how much protection we need in the situation they're in. Les brought up Missouri, and I've got good connections in Missouri. My wife is from there. I know a veterinarian in Missouri. He's not in practice anymore. He tells me, he guarantees me none of his cows have BVD, and most of his neighbors don't. And I kind of looked at him one time and I was there, what are you talking about? How are you sure about that? He said, because every one of my neighbor's cows, that when they get on my property and I have to pin them so the neighbor can come get them, I take an ear notch out of them and sample every one of their cows because I want to know what I'm exposing my herd to. I kind of looked at him. I was there, okay, well, they're on your place. That's one way to do it. But he's that concerned about the exposure from his neighbors that when they crawl through the fence and he pins them, he's testing his neighbor's animals to see what exposure his herd is. And so when we think about this is the pre-breeding area. This is really the management pre-breeding to set animals up correctly. I want to take a few minutes now and move on to during the breeding season. And was mentioned, I've done a lot of work with synchronization protocols, still do a lot of work with synchronization protocols, because every reproductive technology we apply really builds off of us being able to control the estrus cycle. Well, a couple of years ago, there was a lot of buzz, a lot of interest in a new protocol that came out of Missouri, the 7-in-7 protocol. And it uses 4 injections. We start with an injection of prostaglandin. We give an injection of GnRH, another injection of prostaglandin, another injection of GnRH. And the early data showed it worked very well. We compared that to a protocol we had worked on in South Dakota, a shorter protocol that uses the same injections. A shot of GnRH, a shot of prostaglandin, shot of GnRH, or prostaglandin, GnRH, prostaglandin, GnRH again. But these 2 protocols work in opposite methods. This protocol, by giving an injection of prostaglandin when the seeder goes in, we induce a persistent follicle. We turn it over with a shot of GnRH to have these animals ready with a nice growing follicle on day 0 before when we pull the seeder to go into breeding. In this protocol, we try to get rid of persistent follicles. We give an injection of prostaglandin to get progesterone as low as we can, so more animals respond to this GnRH injection to allow better control of follicular development through this phase. And so even though these 2 protocols require the exact same amount of labor, we wanted to see what impact they have. There's actually 27 herds, 1,300 animals. We had herds in South Dakota, Kansas, Arkansas, Texas, New Mexico, Mississippi, and Georgia in this study. When we looked at estrus response, so what animals exhibit estrus, we can see a true benefit to the 7-in-7 protocol. they had more animals that showed estrus after cedar removal. We can also see cyclicity, which I'll get back to cyclicity. Animals that had high levels of progesterone at the start of the protocol had much better estrus response. But what's really critical is pregnancy success. And so when we get down to pregnancy success, we can see cows that showed estrus in every situation. Overall, the majority of animals were bred with conventional semen. We had one herd that used sex sorted semen, but in every situation, those animals that showed estrus had better conception rates. Cycling status, same thing. Those that were cycling before the start of the breeding season, better pregnancy rates. When we get to sex sorted, like I said, there's only one herd there, so our animal numbers get really small, but it's heading the same direction. When we look at our treatments, there's no difference at all. So it didn't matter overall, conventional, or sex semen. These two protocols responded the same way in the area that matters, and that's with pregnancy success. So that's on a fixed time AI protocol. But the fixed time AI protocols don't always fit every situation. And so Lacey Quayle, who's a PhD student with me right now, we were looking at what effort, instead of a synchronization protocol, more of a breeding program. How can we become more profitable? And when we look, the PG 6-day cedar protocol allows us to detect estrus before the cedar ever goes in. And so we ran a study across many different herds that we actually compared heat detecting to a total fixed time AI. And so then it really comes down to, what is your time and labor worth? And she's gone through and we've broken it all apart. She's worked with an economist on modeling a lot of these different things. But when we look at sheer profit, so revenue minus expenses, we can see the 6-day, if you can put the time and labor into heat detecting, actually is more profitable. If you don't have the time or labor to be able to heat detect, we can see the 7-day, a total fixed time AI, did very well. And so we want to have those options available. And that's the reason the Beef Reproductive Task Force has heat detection option, fixed time AI options. Those options are out there both for heifers and for cows for what fits your program. And just like comparing the PG 6-day to the 7 and 7-day, I tell people all of these programs work very good. All of these protocols are going to come out similar when they're run correctly. But we have those options out there. We also have options for sex semen and even adding in more heat detection. So some of you have heard about the split time AI. Basically, instead of doing a true fixed time AI, you do a heat detection and then a cleanup AI instead of just breeding every 12 hours like we do frequently. And so those options allow us different things. The other area I've really spent some time on, I want to briefly touch on, is after the breeding season. And when I was in South Dakota, it was really common that heifers were kept in dry lots throughout the winter. And so when they do that, in the spring, everybody was ready as soon as grass greened up to get them turned out. And what we wanted to look at is heifers that were developed in dry lot situations versus those developed on pasture. They were developed correctly. We can see 97, 94% cycling before the breeding season. But look, we can see a 10% difference in pregnancy rates. And we did several studies looking at it. These animals, when they go out to pasture, they lose weight. They are active. All they're doing is searching for that feed bunk for about the first 7 to 10 days. These animals go out and they start grazing right away. And so we were really interested is, can 6 days of nutrition impact embryo quality? And we can see there's several replicates here. There's about 130 animals. Good embryo recovery rates. Those that post AI for 6 days continued to gain, or those that were restricted to somewhere between 50 to 70% of NRC requirements, we can see embryo stage is decreased, embryo quality is decreased. The one I like to really point out to producers is these embryos had 70 cells into them. These embryos had 49. They were all fixed time AI the same time, the same day. When they walked out of the chute, they went to the 2 different pens, every other animal. And so we can see what nutritional change right after AI does. And if we break it apart further, we can see if we restrict nutrition before the breeding season, before they're AI'd, we impact embryo stage. After AI, we impact embryo quality. And we've also been looking some at the mineral content, and we can see that when diets are decreased post AI or pre AI, we actually change the mineral profile in the uterus that embryo is developing in. So really the biggest thing I like to tell people on nutritional management around the breeding season is we need consistency. We don't want big changes where these animals go through big shifts. And then the last thing I'm just going to barely touch on a little bit, because a lot of this is really still at the lab level, is bull management. Really the most important thing, the most beneficial thing we can still do is the BSE on our bull. We know the return on investment is really great because we can find animals that are infertile. Now, when I talk about BSEs, I tell people we find infertile bulls because these are all bulls that have passed a BSE, came out of a bull stud. We can see differences in conception rates. This was one study just looking at conception rate. There's about 5,000 head bred here, all on Angus bulls. This was a study we ran several years ago. We were looking for sperm longevity. And we can see here, bull A had great fertility all the way through. Bull B, when we bred 18 hours after the onset of estrus, had great fertility, but his sperm longevity wasn't as long. And so that's the direction we're trying to go now, is really looking at the environment that sperm is in. What is impacting it? So we're looking at all the different proteins. And what this really shows you is if you add up all of those different numbers, there's hundreds and hundreds of proteins. So it's a really complex system and it's going to take a lot of time to figure out, but there are several labs working on that area trying to figure out what can affect fertility. The other thing to think about, that when I moved to Texas, they had a model already going here on prenatal stress. And if we stress a cow while she's pregnant, what happens to that calf? Well, Dr. Randall before had looked a lot at the female side, and there was no difference in antral follicle counts. There was no differences in fertility. Well, when I got here, I wanted to look at the male side. And what's interesting is, so these animals are stressed when they're pregnant. The calves are born. They're all managed together. They're all raised together. We let the bulls reach puberty. We collect semen on them. And what's interesting is if we put that semen into an IVF situation, no differences in fertilization rates. But when we get all the way out here to blastocyst rates, those animals that were stressed in utero, the semen they produced had produced fewer blastocysts. When we look at that semen in all the methods we have available, viability, membrane potential, oxidative stress, DNA damage, there's no differences between them. And what we really see, you can see by these color patterns, these are bulls that have good embryo development, bulls with poor embryo development. Their DNA methylations are very different. And so we're actually impacting the genome of that animal while it's in utero. And even the rodent models say a lot of this is passed along the male side. And we can see how it's carried through the sperm, even all the way out after they reach sexual maturity. And so there's a lot of information there. Really enjoy the management side. Like I said, all of the stuff we look at is really looking at management. This is the group that's really critical, all the grad students I've had over the years, because they're the ones that have done all the work. And so want to thank all of them for everything, and want to thank y'all for their time, for your time this evening. And I think we got time for a few questions.
If anybody has any questions, type them in chat. Alan had a comment more than a question. He said, we need a cheat sheet for best practices, and that's probably right, George.
Yeah, several of us have talked about it, but it's getting the time, getting one put together.
Just real quick on the vaccination stuff, what would be your best, your cheat sheet for best practices for the vaccinations?
So our cheat sheet for that is, if you're raising your own replacements, getting a modified live into them around weaning, getting 2 doses of that modified live into them to start priming the immune system. Any of your steer calves that are then being marketed, they've been primed that way too. They're ready to go to market. As your replacement heifers get pre-breeding, we can switch to a killed vaccine, not have the negative consequences of the modified live, and therefore we've got both sides of the immune system, the innate and the active immunity, primed, ready to go. And it gives us a lot more flexibility of when we vaccinate around the breeding season. Here at the research center, we vaccinate with a killed vaccine at preg check time. And part of that is because then we get higher antibodies in the colostrum when we go to calving. And so we're trying to look at it that way, and then we avoid those negative consequences. So let's see, what about dewormers? Dewormers, those studies have been looked at. And if you've noticed, even on the vaccines, I'm talking about the viral vaccines, we have looked at the bacterial vaccines, and we don't see any negative consequences that way. It's just in the viral vaccines. Dewormers, actually, there's a few studies published showing benefits if you get that into them pre-breeding, because you actually increase the nutrition on them. We've got a current study going on here at the research center that we're looking at some resistance. And so proper use of dewormers is critical there, but there's not any negative consequences because you don't really impact the immune system. Like I said, there's a few studies that show benefits because you help the nutrition level by deworming animals. I gotta find where my chat box went here. Here we go. Do vaccines have negative impact on bull reproduction? That's a question we've gotten asked a lot. Nobody has looked at it. With the bulls we develop at the center here, we've talked about it. The question really becomes what measurement we test. We're trying to get, with some of the other studies we're doing on the male side, a better measure of fertility that we can measure in the lab. The studies that I'm aware of on vaccinating bulls have just been on bulls that have been done and turned out. And so we don't have a good enough measure on the male side of if the vaccine affects semen quality, any of the DNA chromatin. We just got those assays up and going this past year. So we're hoping to work into that area, but it's really hard to measure fertility in a bull. And so that's why there's not a lot been done in that area. So I have not seen anything negatively consequence on the bull side. Good protocol on 2-week-old calves. So I'm guessing vaccination protocols. And those, there's controversy on, because you still have your maternal antibodies if you get good passive transfer from your colostrum that don't allow great immune response in young calves. However, there's a study that came out just a couple of years ago that actually showed when they came back and boosted those animals, they had a greater response to the booster even when they had vaccinated that one. And so on young calves, I think a lot more, and what we're trying to look at here at the research center is more of the passive immunity from the colostrum we can get in trying to get those animals set up correctly and then getting them vaccinated prior to weaning. We come in about 30 days prior to weaning with our first dose when they're not stressed, and then give the booster dose at weaning. So why combine modified live with killed vaccines? So on that, as I've mentioned on the heifer development, is really looking at the immune system. And so you have both antibody production, which killed vaccines, when we think about the immune system, there's more to it than just antibody production. You have all of your immune cells. And so when we give a modified live, we let the body recognize how a virus replicates. Because what we do is we give a small dose of a modified live, we let it start replicating in the body. And as it hits a certain threshold, that's when we get our immune response. With a killed vaccine, what we do is they've grown all the virus up in culture, they've chopped it up so it can't replicate or grow in the body, and then we give a huge bolus dose to the animal. And then the animal recognizes those different fragments of the virus and has the immune response that way. And so by those two methods, by combining a modified live early in life when we're not going to have negative consequences on reproduction, and coming back with a killed, we actually allow the body to see more of the immune system or more of the virus and how it operates. And so right now, those are kind of the recommendations for the best overall immunity. I will say here at the research center, we only use killed vaccines, and part of that's for the research we have going on. So then we know the history of the animals when we pull them out of the herd for different studies. And so it gets along fine that way. But if we have high levels of exposure, we're also isolated here. We don't share very many fence lines with neighbors. So I'm not worried about exposure that way. Excuse me. Should we give lepto separate from our viral vaccines in order to better time both vaccines? We can. We did that in a study where we separated. We wanted to see if lepto or vibrio had any impact on reproduction. And so it allows that. We know the duration of antibody production in that is not as long for those as it is for the viral side. And so it really comes down to when your exposure is. With lepto, wet areas, things like that, that we're worried about timing of exposure, we do get better benefits if you fit that timing. And so really that's the one question I get asked a lot. Actually was asked that at our reproductive strategies conference last year, is I always tell people you need to check with your local vet because they know what your local exposure is. You know, I know my exposure here at Overton, but I don't know what all of your neighbors, what your exposure is. And so that's where having your local vet working with you, knowing what the area is and the timing of problems gets to be critical. If you haven't done a vaccine program and are raising ASH-free cattle, would there be any value to starting a vaccine program? Really, with the vaccinations, comes down, they're an insurance policy from exposure. I was talking on this topic at the Range Beef Cow Symposium last month, and there was an individual from Wyoming, he's either from Wyoming or Colorado, brought that up, that he's never vaccinated for anything. In that in that part of the country, there's not a lot of exposure to things. And so if you are isolated that way, then what you worry about is what you bring into your herd. We do recommend vaccinating just because of what the exposures are. How are your animals getting exposed? Do you have fence line contact? Do neighbors' calves crawl through the fence? And so that's where it really comes down again to talking to your local vet to know what your exposure and what your risk factors are. I've worked with herds that have had risk factors everywhere from they have a feedlot and their cow herd and they share a fence line, they go through the same working facilities. That's a totally different situation than somebody that's only a cow calf operation. Or there in Kentucky, you have stocker operations. And so you have to really start to evaluate what your risk level is. See, how early can you give pre-breeding shots and still expect to be effective? So all of the vaccine companies have run duration of immunity out to 12 months of duration. And so we know duration of immunity on our reproductive vaccines is out to at least 12 months. And so exposure over that 12 months, we know we've got them covered. It's not like we give a pre-breeding shot 30 days before and they have better coverage. They have higher titer levels, but it's not that they're not immune. And so that's where the options of getting in and giving cows, we've got that study going on right now with a couple of cooperator herds. We're vaccinating everywhere from 2 weeks before the start of the breeding season. One herd does that all the way out to 6 months before the start of the breeding season. And we're looking at that response through there. And I think we're up to about 3,000 or 4,000 head that we're looking at that away. But we do know the duration of immunity of all the commercial vaccines that are out there is at least 12 months. That's the reason it's recommended to be on annual boosters. None of the pharmaceutical companies have taken duration out past 12 months to see how long after that it'll go. But we do know it's at least good for that. Let's see. Next one, if you've used it, what is your opinion on Multimin 90? It really depends what your mineral program is. So we have worked with herds that have used Multimin. We did not do much of it in South Dakota, partly because Multimin is high in selenium. And in South Dakota, we actually had areas of the state that were already toxic to selenium. And we had people that if they would have used Multimin, it would have put them over the edge and actually had toxicity issues. It does get used a lot more here in Texas, where a lot of people aren't feeding mineral year-round. The studies that I'm aware of that have looked at it, if you are deficient, it does a great job boosting animals for a period of time, and then it starts to dissipate. And so even some of the people I know that have looked at Multimin, it's really timing when you think those animals need more minerals. But if they're on a really good mineral program, I have not seen any data to show that there's big benefits to it. What role does Vibrio play in the breeding protocol? As I mentioned, we did separate out Vibrio and Lepto from the viral vaccines. When we did that, we saw no negative consequences on the Vibrio Lepto side. all of the negative consequences we've seen have been on the viral part, IBR and BVD of the vaccination program. And Sarah's second project that we're working on is actually trying to separate out BVD and IBR to see which component in there might be having a bigger impact. And so, really, the other vaccines we haven't seen much consequence on. It really does seem to be the viral side. And what's interesting is even it crosses species, because with the COVID vaccine, there's a couple of large studies in women that showed even the COVID vaccine, which is a viral vaccine, disrupting estrous cycles for 2 months after the vaccine was used. Oregon Health Science Centers published a study with about 5,000 women in it that showed that. And so really all of our work has moved to focusing on the viral side and the effects of the viral vaccines on the immune system, because the other vaccines we haven't seen negative consequences with. Did I run out of questions, Les?
Yeah, I think you did, George. Want to thank you tremendously for taking the time out of your evening to talk to us. I know this work is a lot of people have been anticipating listening to your program, and we appreciate everything that you have done tonight to help increase our knowledge base, particularly when it comes to vaccination protocols and rebreeding. Before we start sending everybody on their way, I want to remind you that we on February 13th, Dr. Van Valen will be discussing what's the cost of a cheap mineral. I'd love to have everybody join us then too. And Dare, Katie, Jeff, Kevin, if you guys have anything you want to add or comment about George's presentation, let's holler in. But otherwise, George, thank you a ton.
Thank you very much.
Thought
I'll
that
have a
was
wonderful evening.
that was fantastic, George. Sure appreciate it. And as a quick reminder to you guys, next month's Dr. Van Valen's, that will close us out for the year. We've turned this into sort of a winter session deal. So be sure and join us next month for Dr. Van Valen. And again, thanks a bunch, Dr. Perry. That was really, I didn't know you repro guys could actually be practical sometimes. That was great.
Now.
Just a quick reminder, don't forget about the Beef Efficiency Conference on Thursday morning. We'd love to see as many of you out there as we could get you out there for that. It's a good program, a lot of good speakers coming in for that one as well. Great job again, George.